Okay. Hi, how are you? Welcome to the Chromosome 18 Clinical Research Center. I'm Jannine Cody.
Wonderful.
And we are here at the UT Health Science Center. And most people think they're going to drive up to a building that says Chromosome 18 Clinical Research Center, but we're actually a collection of people that have been working together for a long time here at the Health Science Center. We're in the School of Medicine in the Department of Pediatrics. So let me show you around.
Oh, thank you, Jannine.
So we're in the Department of Pediatrics. The offices and labs here are all members of the department. So our office is down this hallway. So here's our office suite here, and it begins with our board with all the pictures of our participants. The families have sent us over the years. We have over 600 families enrolled now. And so Annice created the board, and so we enjoy looking at all of our gorgeous kids here and adults.
Really nice.
So you come this way. Now these office suites here are ours. Here is Annice. If you want to come in. So
hi, how are you? Hello, I'm Annice Hale. I'm the project manager here at the Chromosome 18 Clinical Research Center. It's my privilege to be the first contact for families who have children or adults with chromosome 18 abnormalities. They're looking for answers or more information about our research. With that privilege comes a great responsibility to sign up as many of these families and enroll them in our research as we can. Because chromosome 18 abnormalities are so rare, it's very important that we get as many kiddos and adults in our study as we can, so that'll get us further along in our path to treatments. So I help the families enroll in which includes signing consent forms, sending us the medical records, including the child's or the adult's genetic report that shows their chromosome 18 abnormality, and the last step is giving us blood samples from the kid and from both biological parents if they are available and if they're willing to enroll also. But when these families contact me, I also direct them to the registry if they haven't already found the registry, because I know what that can mean to these families to have the support of their peers. Every day I get mail or phone calls or emails from families, physicians, or even sometimes genetic counselors asking for questions or asking for answers to questions they may have about their child's or their patient's mental or physical health. I try to answer all I can, and I send the rest of them to the healthcare professionals on our team that can answer those questions. When we're looking for very specific information in a subset of our study participants, I'll prepare a survey and we'll send out to those particular families, or sometimes we'll just send them an email and get it. I also make sure that we're in compliance with all the mandates for the ethical conducting of human subject research. When we have dollars for clinical visits, I schedule those families with all the physicians on our team, and I also make all their travel arrangements. You may have been expecting to see a lot of file cabinets here in my office, but we finally entered the 21st century, and all of the files of our study participants are on our computers. I have one small file cabinet that's for the families that are in transition in the enrollment process. And besides working for Jannine, the other best part of my job is meeting all these great chromosome 18 families. Okay, I was going to do this without a Texas accent, but what?
Welcome. This is Chromosome 18 Central, the nerve center for the whole operation. And in this office suite, we have Annice Hale, the project manager, out at the outer office, and Dr. Hale right next door, and Hasi, who you'll talk to in a minute, who's the patient navigator. Across the hall, we have our administrative offices, and you'll meet the people over there. The next office suite down has Bridget Swallow's office, and you'll talk to her. And then right around the corner, we have 3 labs going down the hall, and you'll get a tour of the labs and meet Patty, and she'll show you what she does there. So here is really where I try to coordinate all the different projects. We have at the moment about 6 papers we're working on. We have well over a dozen collaborators all working on various projects from surveys to clinical data. And I try to keep track of everything that's known and becoming known about the 264 genes on chromosome 18, in case any of that information has relevance to anyone with the chromosome 18 abnormalities. So that's why I have lots of piles and stacks and lots of lists, and my lists have lists because I'm keeping track of all those things all the time. Okay.
Go ahead.
Okay, so this is Mimi's office. Let me introduce you to Mimi.
Hi, Mimi. So nice to
Mimi
meet
Hasi.
you.
Hi, my name is Mimi and I work for the research center for about 9 years now. The things I do here are when we have a family visiting our center, I go with the family to all of their appointments, and those appointments are located at different sites of the city. I make sure there are no problems during their appointment and I'm there for them to help. The other thing I do is when we get medical records, I sort, I scan, and abstract medical records. I make sure I update medical records as well. I do assist in preparation of research publication as well. I do collect and analyze data, and I do other tasks as needed.
So I'm Dan Hale. I'm a pediatric endocrinologist at the UT Health Science Center. I got involved with the Chromosome 18 Clinical Research Center in a somewhat unusual way. When I first moved to San Antonio in 1994, my boss at the time told me that my primary responsibility was to get those crazy ladies on the hill off his back. The Hill is a name for the medical school, and the crazy ladies on the hill were Dr. Cody and also the vice chair of the Department of Pediatrics at that time, Dr. Celia Kay. So I started my association in relation with the Chromosome 18 group in 1994, which continues to this time. I play I've actually played 3 major roles with reference to the Chromosome 18 Clinical Research Center. One, I serve as the medical director, and by that, I'm the person that interfaces with other medical professionals locally when we're bringing patients in to do various studies. And I'm sort of the person that would know who is the person that would be a good subspecialist to pull in. For example, if we needed a gastroenterologist, I would know which gastroenterologist would be most amenable to working with our team. The second role that I play is that I am largely responsible for overseeing all the staff that in any way undertake medical procedures, such as research associates. And then the third role, and probably the way that I most frequently interact with families and with their providers, is that our kids come in and our young adults come in from all over the country and indeed all over the world. And I often am the person that is communicating back and forth with their primary care provider or with specialists in their local communities that have little experience with chromosome 18. Fortunately, due to the Chromosome 18 Clinical Research Center, we have probably 50 times more experience than anybody else does in dealing with these children. Most practitioners out in the community may have seen one child with some chromosome 18 abnormality, but very few has seen 200 or 300 children with 18q.
Hello, my name's David. I work for the Chromosome 18 Research Center. Dr. Jannine Cody's my boss. What I do is maintain the computers and the databases that we use to store the records. I also do training for the use of the systems, modify and develop add-ons to the databases, and also monitor the security of the databases and the records that we keep that are all electronic. I'm the troubleshooter for most of the systems here. I also do some support for the Chromosome 18 registry computer systems. And anything else I can do to promote the progress of Chromosome 18.
Nice to meet you.
My name's Bridget Swallow.
Very nice.
So I'm the staff psychometrist, and I have lots of different duties.
Wonderful.
When we have a lot of research questions that we ask, and that determines who we bring in. And when we do bring in families to come visit, I'm usually the first person that they're going to see. And I'm going to do a battery of neurodevelopmental tests on them, whether they're children or adults, doesn't matter.
Okay.
I will administer the tests, I will score them, and I write up the reports. And once I finish the report, I give it to our staff neuropsychologist, and she kind of looks over it just to make sure I got everything right, see if there's anything that she wants put in there. Then we send that to the families. And when we don't have anyone coming in, I have a bunch of different things.
Okay.
I will mail out packets to the families of different types of surveys, behavioral surveys, autism surveys, executive function surveys, for the parents to fill out to send in so we can collect the data longitudinally. So if we have other research questions, that also helps determine who brings in depending on who is fitting the criteria of the question that we're asking. I also will have a medication database that I created. So I have families fill out a survey online, and it comes to me, and I will log all the medicines that their child is taking. So if a family or a doctor will call up here and say, hey, is this drug helpful for children with 18p minus or ring 18 or whatever, I can kind of look it up and just kind of see what the parents say works for them or what doesn't work for them, or I can give them different suggestions. Try this, tell your doctor about this medicine. And I also will collect data for our adult patients or adult participants. When they turn 18, I would like to know what their living arrangements are, their education status, are they married, do they have children, do they have a job, and then I will do it every year after that. I don't start till 18, so when they turn 18, that's kind of when they make it onto my database. And then I'll follow up every year to see what changes.
Wonderful.
So,
That was some great information. Thank you, Bridget. I'm glad that you do all that. Thank you for all that.
Thank you. Nice to meet
It
you.
was so nice to meet you. Have a great day.
You do the same.
Hi, I'm Patty Bird, and I am the lab technician for the Chromosome 18 Research Center. This is one of our labs. This is where I do most of the work processing the blood samples that you have sent. So I'm the person that gets together the blood kit, sends it to you, and then when you send it back, I do the processing. So one of the questions we get sometimes is, why do we need so much blood? Well, because we're doing 3 different things with those blood samples. First, we ask for a green top. It's got a different chemical in it than the yellow tops. With that, I set up a culture in the incubator that goes for 3 days. I add some chemicals to it, harvest it after 3 days so that we get what is called a cell pellet. We've treated the cells in a certain way so that we're really going to be able to see the chromosomes. You can kind of see, if I shake it, well, now it got cloudy, so the cell pellet has been dispersed. I drop that onto a slide and then can use fluorescent dyes to look for different regions on the chromosome. We've got a microscope that we use to analyze it, but this is what the data is going to look like. So we can have areas of the chromosome labeled in different colors so we can tell whether people have deletions, duplications. The scientific name for this is fluorescent in situ hybridization, or FISH. So that's what we do with one of the tubes. With another tube, ideally 2 if I have them, because I want to increase my odds of success, I establish an immortalized cell line. Cells in the blood won't grow forever in the lab. They're called primary cells, and they need to have something done to them so that they will keep growing. We add a virus to the white blood cells. It doesn't impact our experiments at all, but it gives us a permanent cell line or an immortalized cell line. Those cells grow in flasks in the incubator. This is 37 degrees Celsius. And when they're happy, you can actually see them as they grow what's called in suspension. They float around and they form large clumps. Under the microscope, it's going to look like a big cluster of grapes. They're going to be plump and healthy looking. If they look like individual little raisins, that's not good. Now, to grow cells, I have to keep them sterile. To grow the cells, I need to use a biosafety cabinet or a tissue culture hood. And the way it works is it filters the air through HEPA filters to do two things. One is to make sure that the air in the hood is sterile. It doesn't contain any dust, fungi, bacteria, viruses, anything that can get into the flasks. And it also then makes sure that the air that is exhausted out of the hood is sterile, so it protects the environment and the workers. Once I have my hands in the hood, everything in here is considered sterile. The media, which are in the bottles, are sterile. I only open them under the hood. The tissue culture flasks, which have the cells growing in them, are also only opened under the hood. Then when I'm done, I close everything up and can move it back into the incubator or into the refrigerator. So that way, nothing gets contaminated and I have nice, healthy growing cells. So what I do after I have grown up the cells is spin them down and freeze them. And that way we have a ready source for cells, DNA, and RNA. So they're kept in liquid nitrogen, which is extremely cold. It's minus 320 degrees Fahrenheit. And I just want to show you what liquid nitrogen will do. I'm trying to not put my fingers in there. It will do that. So it will freeze a glove. The cells are kept in vials that are put into canes and then kept in the liquid nitrogen, and they are then viable, which means I can thaw them and grow them up pretty much indefinitely. We've got two types of tanks. The small one, the vials are held in a cane like this. We have a larger tank that holds 60 boxes of cells. This one is almost completely filled, and each box looks like this. We freeze 3 vials for each participant so that I can separate them in case a tank falls apart. Because basically it's an extremely expensive thermos, and it works just like that. It keeps the cold beans really cool. So here we are in what is the office and computing lab. So once I get the microarray results, I can analyze them using the software, and this is the image we get. And basically we see all the chromosomes along here. We can zoom in on chromosome 18. This screen is this region here. And the way we interpret this is 2 copies of the chromosome are going to line up right along 0. If you have a shift, you can see this red area is shifted to the left, that's a deletion. If we had a shift to the right, that would be a duplication. So this is a person who has an interstitial deletion of this region of 18q. Now we can zoom in, and I'm not going to show you because it takes a while, the software is a little slow, but we can zoom in and continue to zoom in until we can see almost exactly where this breakpoint region starts. Those are the numbers that we're interested in seeing. A lot of you have probably had microarray done clinically. When we first started doing the chromosome 18 research, microarray wasn't developed yet. But our chromosome microarray has been modified so that it's got a lot of probes along chromosome 18, so we can get a much higher resolution reading than most clinical labs.
My name is Courtney Sebold, and I'm a genetic counselor with the Chromosome 18 Clinical Research Center. Genetic counselors are health professionals that work with families that either have or are at risk to have a genetic condition. So obviously I'm a fairly specialized genetic counselor in that I work primarily with families that have chromosome 18 conditions. You might notice that my office looks a little bit different from the offices of my colleagues at the research center, and that's because I'm actually filming this from my home office in Ohio. My family moved away from the San Antonio area in 2006, and ever since then I've been telecommuting. So when I go to work for the Research Center, I have a couple of different duties. I aid in the collection and analysis of data from the different families of the different chromosome 18 conditions. I also help in the writing of manuscripts for different scientific journals. I speak with families that may have recently received a diagnosis of a chromosome 18 condition and have a lot of questions about the cause or about what to expect. I also speak with families that are interested in possibly participating in the research studies. Lastly, I also write some materials for the Chromosome 18 Registry, and as you all know, that's the advocacy organization that's associated with conditions of chromosome 18. I absolutely love my job working for the Chromosome 18 Clinical Research Center, first and foremost because of the families. Over my 10 years working with this group, I've gotten to know a lot of wonderful families and seen a lot of little kids grow up and a lot of teenagers become adults, and it's been a wonderful experience. I also really enjoy working with my colleagues. I learn something new from them almost every day. So in closing, thanks so much for visiting me in my office, and if you see me at the conference, please stop by and say hello. I'd love to get to meet you. Thank you.